Good morning, everyone. My name is Mihaela. Unfortunately, the author of the abstract and presentation, Arnaut Nicoleta, wasn't able to come today. So today, I'll present our study on HIV matrix protein P17 variants and their link to lymphoma, focusing on next-generation sequencing, or simply NGS, as a novel prognostic tool. This research was founded by an PNR program with a declared no conflict of interest, and this work was done in collaboration between University of Medicine and Pharmacy, Târgu Mureș, Romania, and University of Brescia, Italy. Despite the efficiency of antiretroviral therapy, non-Hodgkin lymphoma remains one of the leading causes of death among people living with HIV. However, the matrix protein P17 plays a crucial role in viral life cycle, but in some individuals, it may undergo some mutations, particularly insertions in C-terminal region, determines production of molecular variants known as VP17s. Studies have shown that those variants are involved in B-cell chronogeneity, determining abnormal B-cell growth, a key step towards lymphoma. So the aim of our study is to develop a novel NGS method to detect those variants in the plasma of people living with HIV, enabling earlier identification of those at higher risk of developing lymphoma. So what did we do? We started with 120 plasma samples from people with HIV, 60 with non-Hodgkin lymphoma and 60 without. The RNA was extracted from 200 microliters of plasma. We then reverse transcribed it and amplified. We focused on 153rd nucleotide region of the P17 gene that covers the typical insertion sites. And this slide summarizes the bioinformatic pipeline. So the raw reads were quality checked and trimmed after they were aligned to the reference of P17 sequence. And lastly, we analyzed for variant calling. For accuracy, we only considered variants that had minimum 100 reads coverage, more than 0.5% minor allele frequency, and a significant co-species was considered as significant only if they had more than 10% of the mapped reads. So as a result, we have seen that 78.2% of all samples had at least one or more amino acid insertions. At this bar graph, you can see the most frequent positions where those amino acid insertions appeared. This heat map offers a visual overview of the distribution of amino acid insertions. The intensity of blue is correlated to frequency of those insertions. As you can see, on position 117 to 118 and position 125 to 126 are the most mutation-prone positions, being the hotspots of the insertions. There were identified around 34 different amino acid insertions, and around 20% of samples carried multiple insertions, mainly double or triple insertions. Looking further at the hotspot positions, 117 to 118 and 125 to 126, we were able to see that at position 117 to 118, the most frequent insertion was alanine-rich one. And on the other hand, on panel B, you can see that at position 125 to 126, the most common one were serine-rich. So these findings suggest that different residues dominate at different positions, suggesting position-specific functional preferences in how the protein tolerates or even exploits these mutations. In conclusion, this data demonstrates that P17 exhibits position-specific preferences for amino acid insertions, particularly positions 117 to 118 and 125 to 126, which may influence its structure, function, and oncogenic potential. We are now working on statistical comparison between the groups with non-Hodgkin lymphoma and without non-Hodgkin lymphoma, and in future we'd like to develop a user-friendly bioinformatic pipeline to streamline NGS data analysis and to define those called oncogenic levels of variants through statistical correlation with patients' clinical history. So with further in vivo validation of those variants with oncogenic significance, this NGS approach could be refined and validated for clinical use and has the potential to become a robust molecular prognostic tool for the early identification of patients with HIV at increased risk of developing lymphoma. Thank you.